Hi students, this is Mr. Eckert. We're going to go through Target C13, which says that you should be able to describe the steps in DNA replication, which of course is how DNA copies itself. So first, I'm sure you know this already, but what does it mean to replicate? If you get a replica of something, if you replicate something, of course you copy it. So when we're talking about DNA replication, it's DNA making a copy of itself, and you know that, or you should know, that happens during interphase, during S of interphase. So S stands for synthesis. When the DNA is synthesized, it's actually copied. So we start with, of course, one double-stranded DNA molecule, and by the time synthesis is finished, we have two identical double-stranded DNA molecules. Remember that synthesis is happening in interphase, and that comes before mitosis. So it is in prophase of mitosis that the chromosomes actually form. So this is actually occurring before chromosomes are forming in cells. So it says if this unit is about the cell cycle and cell division, why is it important to replicate the DNA first? And we may have talked about this already in class, but of course if you have one cell that's going to form two cells, each new cell, each of the daughter cells, is going to need a complete set of DNA. It's going to need a complete copy of DNA. If it doesn't copy before the cell splits into two cells, then there's only two possibilities. One would be that one of the daughter cells is going to get all the DNA and the other daughter cell would get none. Or the other possibility is that they would somehow have to split the DNA, either half, 50-50, or some other ratio. And because the DNA is the master molecule, contains all the genes, all the hereditary information, and all the instructions for how to make proteins, all the proteins, that of course is neither of those scenarios is going to be good. So there's three steps that you should know for DNA replication. It says it looks a lot like transcription. We may or may not have covered transcription yet. If you don't know what that is yet, don't worry about it. But step one, an enzyme, and you should know the name of the enzyme here. So in your notes, put a little caret here. And right here, I want you to write helicase, H-E-L-I-C-A-S-E. So this helicase enzyme unwinds and unzips the DNA. And if you're familiar with the DNA structure, you may even know what exactly the enzyme is breaking in the DNA molecule in order to unwind it and unzip it. And if you think carefully, you may have come up with the right answer, and it's the hydrogen bonds that are found in between the nitrogen bases. So then another enzyme called DNA polymerase attaches to each strand of the DNA. So if you imagine it unwound and unzipped, you can picture that there would be two strands of it. In step two, this DNA polymerase enzyme reads the strand. Now think about what that would mean when it's reading it. What is it actually reading? It's reading the nitrogen bases, the A's and T's and C's and G's. So it reads those. It reads an A, for example, and then it adds the complementary DNA nucleotide. So it would add a T. If it reads a G, it would add a C. If it reads a T, it would add an A. So it does say it's a DNA nucleotide that it's adding. So you may remember that a nucleotide consists of three parts. It's the nitrogen base, a sugar, and a phosphate. So by the time it adds all of these DNA nucleotides to that one strand, then it has the sugar phosphate backbone that is on the other side of the DNA molecule. So that continues until the entire molecule of DNA has been copied, and at the end you have two identical DNA molecules. So here's what an artist envisioned that look like. Here you can see the helicase enzyme that's kind of purplish, and it's breaking those hydrogen bonds in between these nitrogen bases. And then here's a DNA polymerase enzyme, and here's another one. And they're reading the nitrogen base letters, and then they're attaching these nucleotides. Now it looks like it's all one complete strand, but it's attaching it one nucleotide at a time. This type of replication is known as semi-conservative replication. So if you look at this diagram, it's showing, it says old, but I prefer original DNA. So the original DNA molecule is here, And this is how it actually copies, so that you can see in semi-conservative replication, what happens is it keeps an old or original strand and it adds a new strand. So here's the original strand, and then here's the new strand that's been added. That's called semi-conservative. It has conserved one of the original strands, and it uses it as a template to make the new strand. If it was completely conservative, it would have both of the original strands and just copy both of them all at once. So again, that's not the way it works. It's keeping one strand, using it as a template, and then adding it nucleotide by nucleotide to make the new strand. In dispersive replication, you can see here that what it does is it copies bits and pieces at a time from both strands. And so again, you should know for test purposes that it's a semi-conservative replication. And again, that means it's using one of the strands as a template. Now, again, for test purposes, you should also know what the two enzymes are that are involved in replication, and that would be the helicase and the polymerase enzyme, and what those do in those steps that we went over. So if you have any questions, please make sure you ask.